Buenos días. Muchas gracias por esta invitación. Es un placer estar aquí. I'll switch to English given the international nature of this audience. So I'm going to talk to you about generating and shaping novel actions in health and disease. And I will take a little bit of a different approach from what we heard so far. So I will take inspiration from the clinic to go and try to understand how basic mechanisms work in the brain and then go back to the clinic. My presence here was inspired by mutual visits between the Fundación Reina Sofía and our own foundation. This is Su Majestad visiting with our president earlier this year and with Jesus Avila and Miguel Medina. And we really hope that through multinational collaborations in the Iberian Peninsula and other places, we can tackle the problem of the aging brain, which we don't want to admit it, but it is becoming the most important problem for human health. So in terms of what we do, throughout our lives, we learn many actions that are new, that are individual to each of us, and that we use to impact the world, to change things around us. Now, many of these actions, you say, well, you associate a particular stimulus with a particular response, these sensory motor transformations. And these are well studied throughout the history of neuroscience. But many other actions, we actually don't learn in this way. We do, and then we learn the consequences. For example, self-paced actions, actions we initiate by ourselves. There are no external stimuli that can explain them. There may be internal stimuli, but then we learn to do them from the consequences, right? If I hit the microphone, I then learn that I shouldn't do it or that I should do it. But first, I initiate the action. So the concept that we learn from consequence means that first we do a self-paced action, right? So something inside us, we have mechanisms inside our brains that allow us to start something. And then if we encounter something we like, we want to do it again. So we want to retrieve that motor memory and do it again. Now, I think it's very similar for memories, right? We experience something, we store it, and then we want to initiate again that particular memory. The problem of retrieval, which in early Alzheimer's at least, it's so common. So we study this. How do you initiate a self-paced action? How do you then shape it when you know you want to do it and automatize it so you could do it in a wonderful way like that answer over there. Most of what I'm going to talk today will be in this particular species, the mouse, and here's a mouse that has learned to press 8 times that particular lever just based on the fact that there is a lever and every 8th press, a drop of sucrose is delivered over there. So there's no instruction, just generation of action. And now, this generation, the most basic problem of initiating an action, is impaired in this disorder that we just heard at the end of this other disease. So in Parkinson's disease, we have postural problems, tremor, and rigidity and bradykinesia, but we have problems in initiating specifically self-paced actions. If you talk to the neurologists, they'll tell you exactly that there's a deficit there. Now, using these, we can take inspiration. So the mechanism we just heard, we know which neurons die first, or that cause the disease in Parkinson's disease. So we know the problem must be there. So let me use just an example. This is just to elucidate. So imagine this red ball is an animal or a human being, and it's not moving. To make it move, you need a force, right? Something. But when you initiate movement, if there's no inertia, which we certainly have in our muscles, you continue. So to stop it or change direction, you need another force. So what we look at is where are these forces inside our brain? Where are the things that make you initiate, stop, transition actions in a self-paced manner? And when we started to look at these, I'll go back. We developed ways to put accelerometers and very high-speed video cameras in the brain, or on top of the skull and in the body, so that we can distinguish exactly moments of mobility from moments of immobility. So if you see here this bimodal distribution and these videos, what I'm just going to show you is we can detect with great rigor when an animal or a human is immobile, except for small postural changes, and when it initiates movement with millisecond precision. And you're going to see this is going to be important. So if we look in the brain, what's the activity in the brain now of a mice? We can put electrodes inside the brain, extracellular electrodes like it's done in humans, and we can ask what's the activity in different brain areas. If you go to the frontal cortex and the striatum, the main input to the basal ganglia, you find many neurons that just before movement initiation have a profile like that, a phasic increase in activity. And this was somewhat expected. This is what's been reported in primates, even in humans with surgery. But what is thought is that the input from the substantia nigra pars compacta, it's a tonic input that releases dopamine to striatum and frontal cortices, and that's what allows these striatal neurons, for example, to fire. They are very hyperpolarized. When we went and looked at the activity of these neurons, we found this surprising thing. They also had a transient activity just before self-paced movement initiation. Now, this was very surprising to us because we thought maybe it should be more tonic, right? But at the same time, it was exciting because we know that, as I told you, if you have no dopamine, and here's a pharmacogenetic case in which we create a model of lack of dopamine and Parkinson's, you have no EMG activity at all. If you have a lot of dopamine, you're hyperactive. Now, so is it really true that this transient activity exists before movement initiation? To do this, what we did was the following. We put some calcium-sensitive fluorescent proteins, GCAM6F, in dopaminergic neurons in mice of the nigra, and then we put an endoscope, a very tiny endoscope in, and we can here, I think for the first time, image transients of activity of dopaminergic neurons in awake behaving animals. And then we can align to the precise moment of movement initiation here. You see these neurons becoming active just before movement initiation. If we look at all the neurons we record, about 30% of the neurons have transient activity between the self-paced initiation. So no wonder that if you lose more than 70% of these neurons, you have then problems in movement initiation. And furthermore, if you correlate this activity before movement with the vigor of the movement initiation, there's a high correlation. Okay, but is this causal? I mean, is the activity important for initiation? To do this, we do a different type of experiment. Now we put a protein using optogenetics, ARCH-T, that when we shine green light, we can turn off the activity of dopaminergic neurons for 5 seconds only, or for 15 seconds. So now we have precise control this is not a model of Parkinson's, of course, but you can shut it off on demand, and I'll just show you a video of what we can do here. This is a dopaminergic neuron identified, being recorded in the nigra. This is a mouse moving around, and at one point I'm going to shine green light, and you will see the neuron stops being active, right? And the mouse goes to a corner and stays there. And after a few seconds, we stop the light, and you'll see a few interesting things. One is sometimes there's rebound excitation, these little bursts, and the animal initiates movement again. So if we look on average what happens to acceleration when we shine green light in the animals that express the optogenetic protein HT in the nigra, you see less movement, but not in control animals. And you can see here, again, in the bimodal distribution, you see the trials in green, the animals, when the laser is on, the probability of being immobile is increased. Now, the peaks are the same, which means actually the movement quality is not different. It's just the probability of being in the state of moving or not moving. But what we want to ask is really the question of, is it important for initiation? So to do this, we do the following type of experiment. We take trials in which for the 300 milliseconds before we turn the laser on, the animal is stopped. So for 1/3 of a second, the animal is stopped, and then we turn the laser on. What happens? What happens is, if the laser is on, so if the neurons are not active, just during that brief period, the animals don't initiate the movement. If the laser is not active, they eventually initiate the movement. Now, we can do also the converse experiment. If the animal, for 300 milliseconds, already initiated movement, and now we shut down the dopamine neurons, what happens? And what happens is they continue to move, not differently. Now, if you have a good eye, you could say, well, but that green line is going down. Could it be bradykinesia? Could it be that they're getting slower? Or could it be that once they stop spontaneously, they have difficulty initiating again? So we analyze these trials in more detail, and you can see here, and actually there's no difference in the quality of movement until the first stop. Once you're already moving, with this transient manipulation, this is not Parkinson, you continue to move with the same quality until you stop, and then once you stop, you have difficulty initiating again. If something, the latency to stop is increased. Now, can you provoke it? And we can. So if we take now animals that are immobile, stopped, and we just give 4 pulses of light, now with an optogenetic activator, channelrhodopsin, that activate the dopamine neurons. So basically we do at 14 hertz. You see, this is sufficient, this is the blue line here, to initiate overt movement that outlasts the brief period of stimulation. So you just transition into movement. If you're already moving, if something, you reorient and you stop briefly, but then you continue. So this means, in our hands, we thought, it really is that this transient activity is gating initiation. Now, what about learned action sequences? So when you do something self-paced, but then you do something else and something else always in the same sequence, then like the red ball, just the first element is important, right? Just the first element is self-initiated. The rest becomes then an automatic sequence. So to do that, we use the task I showed you, where the animal presses 8 times to get a reward. You see that the behavior becomes very organized, and the animals go from the magazine to press. And now we can ask, what's the activity? And again, you see big activity just before the first press, many neurons active. Now, second, third, or up to final, very few neurons active, and in the final, again active. So the dopaminergic neurons are mostly transiently active at the moments of initiation or then termination, which is a new initiation when the animal finishes and goes to get the food. But so this means that if you've trained an action, and for those of you that see Parkinson's patients, if you train an action, once you initiate it, you actually can carry it over. I mean, I'm not saying in late stages of disease, but okay. Now, can we probe if this is necessary, again, just for initiation or also for performance? So what we do now is we put a little infrared beam on the floor, and when the animal crosses these to start the first press, we inhibit the dopaminergic neurons. And what we see is the latency to initiate goes way up, but not in controls. So now they have difficulty initiating. However, if you do the same inhibition, but after the first press, so immediately after the first press that already initiates the sequence, what you see is actually no difference in the number of presses or the interpress interval, so the animals perform. So what I told you so far is that dopaminergic neurons seem to be phasically active during initiations or self-paced transitions of movement, and they seem to be necessary and sufficient to gate that initiation in corticostriatal circuits, not only in spontaneous but also learned self-paced actions. Now, just for the aficionados, many of these neurons are the same neurons that respond to reward. And so we're working on the hypothesis that these dopamine transients can also set up an eligibility trace to know which actions you should do. But okay, what's the impact of these in the circuit? Now, the substantia nigra compactor neurons project mostly to striatum, to the lateral parts of striatum. And in striatum, I told you, you also find many neurons related to initiation. So if we record the activity of these neurons, you can see here a particular neuron that's mostly just transiently active before the first press of a sequence. So again, related to the initiation. Now, of course, we want to know what the inputs to the substantia nigra are, but we also want to know what's the impact of dopamine in the striatum. And if you take a textbook still today of the basal ganglia, what you read is this. Lack of dopaminergic input leads to less positive activation or modulation of neurons in the direct pathway in the striatum, and less activity and more activity of the indirect pathway that's negatively modulated by dopamine. And because the direct pathway leads to disinhibition of the thalamocortical projection and the indirect leads to overinhibition, you have a balance between prokinetic and antikinetic. So basically, the D1 pathway goes down, the D2 pathway goes up, and you don't move, and vice versa when you move. Now, we have new tools to look at these and probe, so we went and looked. What happens during movement initiation? Is it true that it's mostly the direct pathway promoting it and the indirect pathway not? And to our surprise, and we can do so, again, by recordings of photo-identified neurons or imaging, in this case, in the striatum, the activity of striatopallidal or striatonigral indirect and direct pathways. And to our surprise, a similar percentage of neurons of both pathways were active during initiation. And furthermore, with a very similar latency before the movement. It's not like in our hands one was preceding the other. So both pathways were coactive and then proper movement started. So how can you explain this? The answer to this is still elusive, but I'll tell you just one experiment that reveals a bit this. If we inhibit now the activity of the direct pathway neurons specifically, or the indirect pathway neurons specifically, and we do the same experiment during initiation of the movement, we shut them down. This is the control situation. This here is the light on situation. You see the latency to initiate goes way up. So they have difficulty initiating. But for different reasons why. And I'll summarize here why. In the case of the direct pathway inhibition, the animals mostly stop the initiation. And then when you release the inhibition, they continue. These are videos superimposed with the laser off and laser on. And this is a quantification if the animals leave this zone. In the indirect pathway case, when you inhibit in many trials, you abort the initiation. So what does this mean? So what we've been working, when this type of model was proposed, the hyperkinetic, antikinetic, in the same studies, but maybe it gained less attention, in the same discussions, there was another model proposed, which is more an action selection type of model. And in this action selection type of model, what people have proposed is both pathways are actually coactive, but the direct pathway is important to select what you want to do, and the indirect pathway to inhibit in that context what you don't want to do. And both are needed for seamless initiation. And so we have a lot of evidence that I will not tell you, but we think something like this may be going on in the initiation of self-paced actions. Now, one would say, okay, what about those well-trained actions that you do all your life, and it's no longer self-paced, right? What if you do a sports, for example? What if you run in the treadmill all your life? So here's what happens to an animal that's naive, and where I've blocked all dopamine receptors, and I'm going to place this animal in a treadmill. So this is, of course, an acute Parkinsonism state. You see the animals placed there. I'm going to turn on this accelerating rotor rod. This is actually an experiment I did, still in the time when I did experiments. But, and you see the animal falls. It falls with all four paws down. If you put them with belly up, they'll turn around. You put them in a beaker of water, they swim. There's no muscular problem. There's a problem of initiation. So what happens if you train these animals so much that now when the rotor rod moves, that's a signal to start moving, and they learn this. So we train them for 2 weeks off drug, put them back, or different groups of animals, still no self-paced movement initiation. We place them on, and start the rotor rod, and now they move. So what happened between here and there? You could say, well, maybe the dopamine receptors were internalized. We measured a little bit the D2 were internalized, but that didn't explain the data. What seemed to explain was the following. We saw something with training akin to long-term potentiation happening at the cortical striatal synapse. There was increase in both AMPA and NMDA receptors, so an NMDA-dependent type of plasticity that even resulted in morphological changes. And at the same time, we also saw that the trial-to-trial variability in the activity in motor cortex and striatum, this is an exoteric measure, but decreased with training, meaning the activity became more stereotypical, just as the behavior became more stereotypical. So this leads us to hypothesize very directly, well, maybe plasticity at the cortical striatal synapse is what's you use to automatize and stereotype movements, and then you need dopamine less, because the threshold after that LTP-like induction, the threshold to fire in the absence of dopamine goes way less. Okay, so I'll just show you fast one thing. These, probably many of you have seen. Is this relevant, what I'm telling you, for the disease? This is a Parkinson's patient in Holland with a caretaker, and he's been asked to start stepping. You see there's a touch in the foot, a stimulus, a sensory stimulus, because it's hard for the patient to actually initiate in a self-paced manner. The caretaker touches the foot. Eventually, you start initiating. Then you have these fast movements, sometimes difficulty in stopping. This is characteristic. Now, this patient lives in Holland. And what do people do in Holland all their lives? This came right in the same year that we were publishing this other study. They ride bikes. So a friend of mine from the hospital in Holland called me and said, you have to see these. This is the patient without drug outside the hospital, placed on a bike. Many of you know this video. And when I saw these, I was like, what's going to happen now? I mean, there's a curve at the end, right? But what's amazing is the patient does turn around, does come back, then I was thinking, well, can the patient stop the bike? And apparently yes, right? Jumps off. And now the caretaker says, can you please walk towards the camera? Right? And again, difficulty. Now, I'll mention a bit more towards the end of the talk, but obviously we're interested in how this impacts Parkinson's disease. We used here a trick. The treadmill and the bicycle are a trick because once you start, you get constant feedback, so there's no other initiation. And with Jose Obeso and Ledia, when I presented these results at a meeting, Jose Obeso has had the hypothesis for many years that these lateral nigral neurons are super active when you initiate automatized things, habits, and that actually this makes them, these in humans, even more than rodents, are metabolically active neurons that project all over the striatum. So now we're actually together trying to test Jose's hypothesis in mice and then back in Spain in primates and humans that indeed the overactivity of these neurons in the most habitual initiation of a lot of the things we do in everyday life can actually contribute to disease. But I give credit to him on the idea. Okay, so is the plasticity really necessary for you to select and do whatever you want to do? And so to do this, of course, the rotor rod is not a very good task. So we go back to this operant lever press 8 times, and you see here again, these are all the presses and the head entries plotted in a graph. You don't need to pay a lot of attention, but I don't know if you notice, the behavior becomes very organized after 6-12 days. So the animals do press about 8 times, and they do so faster. But I want to draw your attention to the bottom parts here, these lines going out. This is the coefficient of variation of the movement from trial to trial. So it's, in other words, it's a measure of stereotypy, how equal you are to yourself. And you see it decreases, meaning the movements are becoming more equal. You're selecting a particular pattern as you train. You don't do all the things, you do just one thing. If we use a particular Cre line, the RGS-L9 Cre, and we delete NMDA receptors just from the medium spiny neurons, so there's not this corticostriatal plasticity, I don't know if you can see the animals press, but their behavior never becomes organized. Here in red are the mutants, in blue are the control lead termates, and the mutants do not become more stereotyped. So they cannot select the action patterns that they want. But if we go back to what I was saying, well, what are the action patterns you want? These must be encoded in initiations in the brain. So it means you need them to select the neural patterns you want, right? Because we don't select muscular activity. We must select the brain activity that leads to that muscular activity. So can we do that directly? And the answer is yes. We can place electrodes in the motor cortex and now just say, from the activity in the motor cortex, let's put that activity controlling an auditory cursor directly. So if you are doing the experiment, you see the activity in motor cortex of many neurons, and you hear a tone, wah, 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 wah. And if the tone goes very up, this is an arbitrary activity. If it goes high in pitch, you get a drop of sucrose from the sky. If it goes down, whoa, you get a pellet. Animals learn to do this very quickly. So now, and they stop moving, in fact. They can select the motor cortex patterns that they need to get reward. So directly. And well above chance. And again, if we do the same mutation, they cannot do it. They cannot select the appropriate motor cortex pattern. So this tells us that really this plasticity is about selecting the motor plans you want at the behavioral level, but at the neural level. And just to finalize, of course, we couldn't resist but say, well, then this can be used in humans, so then you can learn to use your neural activity to control anything arbitrary. You just need to give feedback to the neural activity, for example, in this case of a plane controller, and then give feedback to the user, and the user can learn to control a plane. Is it true? So this is Nuno Loureiro in Diamond 42 in Munich, flying the plane just with EEG activity based on the same principles, and he needs to land over there somewhere, so he's turning now. And so basically these are results of Nuno being instructed to turn left, right, and you see he can follow pretty well just with neural activity. And this memory is consolidated. Now, we're not interested in moving planes. It would be very interesting if people that can no longer move, I mean, I'm all for first trying to prevent the neurons from dying, but if we cannot, maybe we can give access to some very simple thing like turn on email, move a wheelchair forward or backwards. So what I told you today is, I hope, for action generation that transients of dopamine activity seem to be necessary to gate the initiation, and they seem to be necessary and sufficient, but once you have very well-trained actions, dopamine is less dependent, and they tend to select what you want. Activity in basal ganglia subcircuits is important, and plasticity, and the way it's organized is more, it seems to be more a selection of the patterns you want, and that this allows you to select the motor and neural patterns you want. And perhaps this can be used to train people to control other devices like we learn to control our body. And I just want to thank the people in the lab, especially Joaquin and Fatuel for these experiments, and the collaborators in these particular experiments, and I hope in the next meetings Jose Obeso and Ledia can present new exciting results with using, as we heard in the last talk, alpha-synuclein extracts or virus to cause degeneration, and imaging, we hope, for the first time, the dopamine neuron activity as they die in a live animal, and see how it progresses according to Jose's hypothesis. Thank you so much for this invitation, and I hope it was interesting.